This Week in Virology, the podcast about viruses, the kind that make you sick.
Hello, Pitt. From Microbe TV, this is TWiV, This Week in Virology. This is episode 373, and today is January 21st, 2016. Hi, everybody. I'm Vincent and you're listening to TWiV, the podcast all about viruses. Today, I am coming to you from Pittsburgh, Pennsylvania. I'm at the University of Pittsburgh. You can be loud, you can make noise, it's okay. At the medical center, and I've been invited here by the Department of Microbiology and Molecular Genetics. I'm actually here to give a lecture in honor of my guest, who is a distinguished service professor here in the department, Julius Youngner. Welcome to the show.
Thank you.
Could I call you Julie?
Of course, everybody does.
Yeah, why couldn't I? Yeah, Julie. So I'm giving the Julius Youngner lecture tomorrow. Should be fun. And it'll be different, so you should come because it's not going to be your usual kind of talk. But today we're going to find out all about Julius. But first, we do have a great audience, which you heard earlier. Thank you very much. We have an overflow. We have another room full of people, and you needed tickets to get to this, right? Did you ever do something where you needed a ticket to get in? I bet not.
Originally, yes. I had
What's that?
A ticket. I had my CV was my ticket.
Okay. Let's find out what the weather here. So when I flew in this morning, the pilot said it's warmer here than it is in Newark, but he was wrong. It was actually sunny and just a few degrees above freezing in Newark, and it's really cold here. Inside this building, the internet doesn't work, unfortunately. What's the weather? Does anyone know? Is it freezing? There are no windows in this room either, so we can't look out. It's probably below freezing, I bet. And it's gray, and it's about to snow, so I might not get out of here on Saturday. 25 Fahrenheit? What is it in Celsius? That's what we have to know.
-4.
Thank you. Very good. And cloudy, I think, and pretty cold out there. I guess that's typical winter in Pittsburgh, right? Yeah, okay. It's my first visit to Pittsburgh. I've been to Pennsylvania. I live in New Jersey, so I've been all over Pennsylvania, but never this far west. So this is an episode dedicated to you, Julie.
Thank you.
We're going to find out all about, here's what we're going to do. We're going to find out all about where you were born and raised and educated, what brought you here to Pitt, what you did here, and a few other things I'll spring at the end. Okay?
Yes.
Julius, I was talking to him before, and I said, we shouldn't really talk because I don't like to talk before a podcast. We just talk it out, and then when you go to do it, it sounds wooden. And he said, don't worry, I have so much to say, it won't be a problem. So we'll see, right? We'll see. So where are you from? Where did you start?
I was born in New York City.
All right.
That's right.
At least one other person here was born in New York.
Somebody.
In Manhattan?
I was born in Manhattan,
Wow.
Lower Manhattan.
Mm-hmm.
And
You grew up there?
I spent the first 18 years of my life in New York.
And so that would be high school, right? Through high school?
Through college.
You went to college in New York?
Yes.
Where?
NYU. University Heights when it was NYU.
No, it was Uptown,
Uptown.
which is now, it's down by Washington Square, right? Okay. What did you major in?
English. My native language.
What did you plan to do with a major in English? Did you have any idea?
Read books,
Yeah.
write novels. I had all kinds of plans, which I soon, as I became more mature, gave up. Realized I was going to have to look for something else to do.
Mm-hmm. So when you graduated, the day you graduated, what did you do?
I graduated from NYU, and I went to the University of Michigan. Mm-hmm. And started a graduate program there.
In English?
No, in what was then called bacteriology. There was no microbiology till much later, but it was the Department of Bacteriology. Now, you may ask, why did I change so abruptly my career?
That would be a good question, yeah.
I thought you might ask that. When I was growing up, I had every childhood disease imaginable. I was a very sickly child. I had periods of good health and periods of terrible health with all infectious disease, especially when I was 7 years old and had pneumococcal pneumonia in both lungs and had to have a I had a drain, I had pleurisy, I had the whole works. This was before antibiotics, there was nothing they could do. And I went through, actually, through the crisis. You know about the crisis in pneumonia?
No.
In the old days before penicillin, patients with pneumonia would be toxic. And there would come a period when the antibody overwhelmed the toxin. And they, almost within hours, the temperature would drop and they would be okay. And that was called a crisis. And I remember bothering my mother very much by calling her to my bedside and saying, I'm going to die. And that didn't make her feel very good. I said, I'm going to die now, Mother. And a few hours later, I was perfectly well. But I had pleurisy, so I had to have a drainage. And it was, don't ask me the rest. It was pretty awful.
So you had many infectious diseases.
I had many infectious diseases.
And this made you think, I want to go to a Department of Bacteriology and work on them?
Yes.
Really? During college or after you graduated?
After I graduated, I decided, what did I want to do? And I decided to go to the University of Michigan because a family doctor, had been there and pointed me in that direction.
Was the chair Frederick Novy?
Frederick Novy? And I said, 1 year I gave the Frederick Novy lecture. And I'm old enough, so I remember as a graduate student seeing him raking leaves in his yard. He was still alive. So actually, I'm a grandson of Robert Koch.
Grandson?
Great-grandson.
Great-grandson.
Great-grandson of Robert Koch.
Really?
Yeah, because it was Koch to Novy, Novy to...
Oh, scientifically a great-grandson. I see, okay.
No, I don't have any Czech blood in me as far as I know. No, I'm a scientific great-grandson.
Got it, right.
It was a direct line.
Am I related to you?
6 degrees of separation, I bet.
So we actually have two things in common. We worked on the same virus,
right,
polio, and we were both presidents of the American Society for Virology.
At widely different times.
Okay, you can point that out, that's fine. I was about to say that maybe the fact that you had all these infections is why you're so long-lived.
Well, you know, it occurred to me, too, that, that really my immune system, if anything would have boosted it, that was it. So maybe my immune system has prospered on the basis of all those infectious diseases that they prevent now from having naturally. I had them all naturally.
So you went to Michigan, and by the way, we did a recent TWiV. I visited Michigan for a ceremony a few months ago, and I learned about Frederick Novy. He had a remarkable career. He actually came out of retirement to discover a virus of rats that he had found and lost. And in the 50s, he came out of retirement to identify it. It turned out to be a parvovirus of rats. Fascinating story, and we did a recent TWiV about it. When you went to Michigan, what did you go to work on?
I went to work on pneumococcal, pneumococcal polysaccharide.
And you got a PhD there?
Got a PhD there.
Whose lab was that in?
Walter Nungester was the man.
Were there any, aside from Novy, were there any virologists there at the time?
Virology, there wasn't even a course in virology. At that time, virology was in the backwaters of, there was no journal of virology. There was, it was a new and upcoming part of what became microbiology and virology.
So just for reference, what decade are we talking about here? The 30s, 40s?
Of what? When virology?
1900s, when virology was still a backwater compared to microbiology or bacteriology. 40s? Okay.
40s.
All right. So you got a PhD doing pneumococcus, and then what was the next step in your career?
Army. The U.S. Army. It was World War II, and I was drafted into the Army and went through basic training. And at the end of the 8-week basic training, we were all shipped out, except when the roll was called, my name was not called. And 99 men were loaded on trucks and taken away. This was the time of the Battle of the Bulge, and they needed any bodies that they could take, even with 8 weeks training. And so I was standing there, and I asked the sergeant, why, there was a camaraderie, a band of brothers effect, even in 8 weeks. And I felt terrible not being with these other guys. And I asked the sergeant, and he said, I don't know. Just go and wait for something to happen. And I went back in the barracks, and I had to, for the next few days, cadge food at mess halls and other units around the camp. This was in Texas, in Camp Barkley, Texas, where the 5th Armored Division of Patton had trained. And a few days later, somebody came with a Jeep and took me to a headquarters, and they told me to pack all my stuff up, and here's an envelope, and open it when you get to the train station at such a time. And when I got to the train station, I opened the envelope, and it said, you're under orders. Nobody can ask you where you're going, but you're going to Knoxville, Tennessee. And so I got on this train, and of course the MPs would come through because they wanted to see if anybody's going AWOL. And I showed them this piece of paper, and they didn't ask me anything. But I asked people on the train, there were a lot of officers and other people. I was a private at this time, and I asked the officers, do you know of a base in Knoxville, Tennessee? They said, nobody ever heard of anything. Well, I get to Knoxville 2 o'clock in the morning, and I had a phone number to call. And I called the phone number, and somebody says, hello. And I said, my name is Youngner, Private Youngner, and I'm supposed to call this number. And they said, where are you? I said, I'm at the train station. They said, we'll be there to pick you up. And I lay down on my duffel bag with my head on my duffel bag and fell asleep. Soon some guy in a Mackinac wakes me up and says, come with me. We got in a car, and I said, where are we going? He said, you're going to a place called Los Alamos. And we got there, and I had to go through fingerprinting, photographing, and then it was the middle of the night, so they said, there's a barracks here where there are other GIs. You go in there, and there's a bed for you. Well, I went in there, but it was absolutely dark. And I was puzzled what I was doing there. I had no idea. And I didn't know where the bed was, so I kept moving through one bed after another, feeling the beds. And of course, I got some very strange reactions. And finally found the bed, and I was so discombobulated that I didn't even take off my shoes. I just lay down on the bed with my clothes on. And in the morning, I woke up, and there were a whole bunch of GIs who were there. And I was there for 2 weeks, in which time I was vetted by the FBI for clearance. And the funny story is that they went to the superintendent of the apartment building on West End Avenue and 86th Street that we were living in. And they went to the superintendent and asked him all kinds of questions about me, whatever he asked. The superintendent then made the mistake of going up to my mother and telling her that the FBI was there. Asking questions about me, and my mother said, I thought, I know exactly what it is. It's because he's a communist. Well, the reason she thought I was a communist is because I subscribed to a newspaper that was a left-wing newspaper. It wasn't, the Daily Worker. It was just a left-wing newspaper that now is extinct. But she was very conservative in her political views, and she was disturbed that from college on I read this paper, and she considered it that I must be a communist, and that's why they're after me. So after 2 weeks, I had the same procedure. I was given an envelope and told to take a train at a certain time, and I was under orders, so I didn't have to tell anybody where I was going, and I went to Rochester, New York, in the middle of a blizzard, and got there and found out that I was going to be working in a group that was testing the effect of inhaled uranium salts on rabbits and beagles, because the workers in the purification plants were inhaling lots of uranium dust, and they wanted to know what level was safe, at least in animals. And so I learned a lot of pathology because we had to do the pathology, and somebody taught me, the captain who ran the 12 GIs who were there, and there were 170 civilians. The 12 GIs did all the dirty work. So I was, one thing I learned was pathology because I was taught by Captain Metcalf.
You had to make sections and look at them, interpret them in the microscope. Right?
With microscope and assess the damage or normal, whatever happened.
And they decided to have you do this because you were trained as a scientist,
That's right.
right?
That's right.
And did you stay there for the duration of the war?
Yeah, more than that. The war ended. Well, it's a strange story because the war ended and the GIs all said, well, now we're going to be mustered out. And they said, no, you're not. And you're going to be taken to Fort Dix. You're going to go to Fort Dix in New Jersey, and you're going to be put into the active reserve. And we said, well, what if we don't want to do it? Because are we going to get salaries that the 170 other workers are? And they said, no, you're going to just get your GI pay. And they mustered us out. But before we went, one guy said, let's get together. We got together and we said, what if we refuse to do this? Because they're taking us out of uniform, but there's no benefit for us. So one guy said, well, I'm the only the unmarried guy here. So what I'm going to do is say I refuse. So he went to the captain, and the next day he was gone. And a few months later, we got a postcard from the Aleutian Islands from him. The thing is that I didn't know exactly what we were doing. I knew something about, we couldn't figure out. GIs didn't know. The secrecy was unbelievable. At Oak Ridge, we were told that this is a project that if we succeed before the Germans, we'll win the war. That's how important this is. And so we took it very seriously. But I didn't know what these things these rabbits and dogs were getting was. And one day I had to go ask a question of a biochemist in that section of the establishment. And he wasn't in his office, but there were a pile of books from the library, and in them was a marker in every book. And books from the library are not secret. So I opened every book and looked, And every book was on toxicity of uranium salts. And I went back and told, this is uranium salts we're working on. And then we had these fantasies of what could happen. It had enough physics, but that time there had been, Lisa Meitner had been, had the first fission experiment. And so we knew it was something about energy, atomic energy from the atom. And we thought, is this going to be driving airplanes, or is it going to be submarines, or is it going to be a propellant? None of us thought of the horror of an atomic bomb. Not one of us considered the possibility that this would be the end game for Manhattan Project. And in August 6th of 1945, I was still in the Army, came back from a visit to my in-laws in Detroit on a train to Rochester, arrived in the station, and the newspapers all said that what had happened in Hiroshima. And so it's a strange life that I've led, because in the 20th century, I was involved in one of the major events that was used to kill people. And later on, within a decade, I was involved in a project that was designed to save lives. So there was this dichotomy in my feelings about the horrors of Hiroshima and Nagasaki, and finally, when the vaccine was finally licensed and used, and all the lives that were saved, it was just like a separation of the two parts of the 20th century for me.
When did you get out of the clutches of the Army?
I got out in 1946. And I went back to the University of Michigan because they had to take me back for 1 year, because I had been an instructor before I left, because they had shortage of everybody. And I was looking around for some place to go after a year, and I heard that the Public Health Service was opening up for the first time commission officers could be a commission officer in the Commission Corps of the Public Health Service.
Right.
And so I applied to the Public Health Service for commission because I wanted to work in infectious diseases. I was one of the first scientist officers, as they called us, in contrast to the medical officers or the veterinary officers. So I went in the same war from being a private in the Army to lieutenant junior grade in the Navy, because the Public Health Service was part of the Navy until '53, when Eisenhower finally declared the end of World War II. It wasn't until '53 that we were under wartime footing, which gave the president a lot of power that he could. That's why they didn't end the war.
So when you got this position in the Navy, Public Health Service, where did you go?
I went to NIH.
NIH, okay.
I was assigned to NIH, and I was assigned against my wishes to the Cancer Institute. And there I worked on the effects of blood supply on tumors for almost 2 years. Lived in Maryland, and it was easy commute from Maryland in those days to the NIH.
Certainly not easy today.
Today, it's a slog.
But cancer is not what you wanted to work on, right?
It wasn't. But what I wanted to do was cell culture, because by this time, Anders, Weller, and Robbins' paper had appeared. And so I was fascinated with cell culture and the possibilities of working with viruses. But before that, a colleague of mine there, who was a senior person there, named Clifford Grobstein, who later became the first dean of the medical school at La Jolla, And he and I set up an experiment which was so simple-minded when you think back on it. It's so simple. It was published in Science, so that it was accepted by the powers that be at the time. But it was one of the most simple-minded experiments you can imagine. And that was to take cells from two different species and grow them in the same culture and see if they would get along. And we did this paper between chick and mouse cells, using pieces of tissue and letting them grow out and grow together, and they were happy as clams. I mean,
So they became clams after putting.
No, they didn't become clams. They were happy at proverbial clam. But that was just a simple, looking back on it, but that was how little was known about cell culture.
At the time, there were no cell lines, right?
There were no cell lines. These were from primary tissues. There was only one cell line. It was the L cell line that was growing, the product of work at the Cancer Institute.
Okay.
And
but no human cell lines, right?
no human cell lines. And in order to do any more culture, I would have had to do it the way they were doing it at the Cancer Institute. And that is the way that was the man at Rockefeller, his name escapes me now, who started cell culture, but in a stainless steel room that was sterilized by steam. And then everybody had to go through a door that was kept a safe door and put on sterile things with masks and hoods, and then they worked inside this place. And this is because the cell culture that was started at the Rockefeller Institute, one of the students who came there and became head of a cell culture unit at the Cancer Institute kept the same thing. And I said I wanted to work with viruses and cancer. And the furor that this caused to bring viruses into this situation was impossible. So being a commissioned officer, I went to the scientific director and told him my problem, that I couldn't do anything there. And he said, this was Harry Eagle. I don't know whether the name means anything to you.
Absolutely.
Harry Eagle was scientific director of the Cancer Institute at the time, and he was interested in cell culture, but because this time he was doing Eagle's solution.
Right.
He was interested in growing spirochetes at the time. He became first dean of the Albert Einstein College of Medicine.
Right.
He said, "Well, Julie, why don't you find some place to go and do this? They're building the new hospital, big hospital, and it's going to have research units in it. And the Cancer Institute will probably move there. And when after you find somebody, go and find somewhere you can do what you want, and then come back." And so I started to look around and ask where I could go. But I had an acquaintance in Ann Arbor from my days in Ann Arbor who I told about this, and it happened to be that he was also acquaintance of Jonas Salk. And Jonas Salk, although we had been there at the same time, overlapped. He was an assistant professor in the public health school, and I was a graduate student. But we never met and never knew about one another. And so this mutual acquaintance told Jonas that I was looking, and he knew what I wanted to do. And Jonas wrote to me, and he came to the community where we were living in Maryland, and we talked, And then we talked on the phone, and then he said, well...
Send text messages to each other? No.
Smoke, by smoke.
No, smoke.
The smoke was the preferred method then. But he said, you're exactly the person I'm looking for who starts cell culture, because then they work only with monkeys.
He was already working on polio at the time, right, when you talked with him? Is that correct?
He was.
He was, yeah.
Yeah, he was working on typing at the time. He had gotten a grant from the National Foundation, and he and 2 other labs around the country were given 23 strains from all over to type them in monkeys to see whether there was more than 3 types of polio.
Okay.
Because if there had been more than 3 types, making a vaccine would have been very difficult. With something like the pneumococcus I suffered from, or the cold virus, rhinovirus.
Sure.
So there were only three types, and we established that. But I was working on helping with monkeys, and that was a terrible chore to type everything in monkeys, because you had to infect them, then make antiserum against each, then test them against the other ones.
So the testing of the serum was done in another monkey, right?
Well, another monkey.
Because you didn't have cell culture to do that in at the time.
Everything had to be done in monkeys, because there was no other host.
Right.
So I worked on that, helping with the monkey project, but at the same time in the lab, I was starting to try to find out what kind of cell culture and learn something about how to do it.
So you've moved at this point. You've gone somewhere.
I went to Pittsburgh.
Right here. Well, not right here.
No, not right
but
here.
this institution, yes.
Up the hill, over the river, and over the river and up the hill.
So you came to an agreement with Salk. You said, why don't you join me at Pittsburgh?
Right. And I was supposed to, after 2 years, go back to...
I see.
To go back to the NIH.
Got it. So you were part of his group initially. Is that correct?
No, there were already three professionals working already, and I was the 4th.
Okay. So were you an assistant professor, essentially?
I was an assistant professor.
Okay.
Right.
So you worked on the monkey project.
I worked on the monkey project, but at the same time, in the spare time, I was trying to set up a cell culture method.
What year is this now, 50?
I got here in 49.
Okay.
And by 51, I had a cell culture system for polio going.
What was the cell culture?
It was monkey testis.
Okay. Now, how did you get to that particular tissue rather than kidney?
Because there were monkeys around that were being sacrificed.
Okay.
And it was a tissue that
didn't want.
Other people had grown.
Why didn't you use kidneys, for example?
Then I went, well, because monkey testes were not a very good, didn't have a very good yield of virus.
Kidneys.
Monkey testis first.
Did not have a good yield of virus. Okay.
So we went to monkey kidney.
Okay. Got it.
And at that time, I found out that pathologists had used trypsin to disperse tissues. And so instead of using pieces of tissue to grow, I found that by trypsinization, if you take the cortex of the kidney, chop it up, put it in a trypsin solution, dispersed it beautifully. And then if you put it into solid surface, glass surface, it would form monolayers. And these monolayers, you could make a lot from one monkey. And so, and the yields were very high. So the next step was, already we had evidence that even from the monkey testes, that if you inactivate them with formalin, the poliovirus, they would immunize monkeys. But the poliovirus produced so much more virus, the monkey kidney produced so much more virus, that was the preferred system, and the rest is history.
So you said that pathologists taught you about the use of trypsin. What did they use it for? To disperse tissues?
Yeah, to disperse tissues for whatever microscopic reasons.
And this you learned back in the Army, right? You said?
No.
After that?
No. This was from my reading.
Reading,
Talking,
okay.
Yes.
But no one had thought to use that to make cells for cell culture.
No, nobody had used that method before. Renato Del Becco, at the same time, I think had done the placking of poliovirus
Right.
in monolayers.
Sure.
So
That was 51, I think.
Yeah,
ish,
so
right?
it was a little bit later.
So did you publish this observation at some point?
Yes.
And then everyone started using trypsin to make cell cultures.
Yes, and as a matter of fact, remember there was a citation index?
Yeah.
Well, in 1960s, I was on the top citation index because of a technique of trypsinization and standardizing how to measure the cells and disperse.
Today you would have a lot of Twitter followers if you did that.
I don't have Twitter.
Yeah, I know. But if you did the trypsin today, you would. So when HeLa cells came along in 1951, I think,
Yes.
immediately they were using trypsin to, in fact, Gay probably used trypsin to disperse the tumor, right?
I had nothing to do with it.
I know, but I'm just wondering, at some point HeLa cells were dispersed by trypsin, which you had developed.
But we didn't use HeLa cells, which were available, because we knew that it was a tumor cell.
Right.
It would be unacceptable to make a vaccine in a tumor cell.
Sure.
So HeLa cells were never considered to make a vaccine.
Did you ever use the HeLa cells for typing experiments?
No.
Or to take serum from the immunization trials to look at neutralizations and so forth?
No, we did it all in monkey kidneys.
All in monkeys?
All in monkey kidneys.
So these were primary monkey kidneys?
Primary monkey kidney cells grown. You could use a secondary growth, but after the secondary growth, they didn't grow anymore.
So then,
poor.
when you were starting to grow batches of vaccine of polio for vaccine purposes, they were grown in monkey kidney cells.
That's right.
As well.
That's right.
And in fact, until not too long ago, they're still produced in primary monkey kidney cells.
That's
Even the Sabin vaccines are grown in those cells.
Exactly. And now it's all a human primary cell line.
Yeah, MRC5, for example.
MRC5.
So I guess you never went back to NIH, NCI.
No, but they wanted me to come back.
Uh-huh.
I was time to come back, but I realized by this time that we were well on our way to getting a vaccine. And I was not going to give up that opportunity to be part of the group that did this. So I switched from active to inactive reserve.
Okay. And you stayed here and worked with Salk on the vaccine project,
Yes.
right? And this trial was in 54, is that right?
The big trial.
But you did smaller ones before that, correct?
We did smaller ones locally, yes. We did phase 1, what they would call phase 2 trials locally. The phase 3 trial was the big trial in 1954, the results of which came out in 1955.
Right.
That was 800,000 children were involved in 12 different states. And there'll never be another trial ever like this.
That's right.
It was a unique event.
Did you ever inject yourself with the vaccine?
Oh yes.
Yeah.
And my kids.
Yeah.
With homegrown stuff, not with commercially produced stuff.
So this is stuff made in your lab, right?
Yeah, because if we were going to go out and do school children,
Yeah.
how could we say we didn't inject our own children? And so my son and my daughter both got the shot.
It's interesting because the Salks did the same with his family, and Sabin did the same with his family. Many investigators who developed vaccines in the 50s did that. It was the practice.
Yes.
Not anymore.
No, I don't think so.
Not allowed to do that, yeah.
No.
Things have changed. So after the vaccine trials, you remained here.
Yes. I remained in the virus research laboratory, which was in what's a dental school now, pharmacy school building. It was called Municipal Hospital at the time. It was a hospital was built for infectious diseases, run by the city, a city hospital called for infectious diseases at a time when antibiotics came out. And it made it a white elephant because antibiotics took care and their population density was so low that the hospital was given over to whatever anybody wanted to do.
Right.
And the first, we had part of the first and second floor in the basement on one wing. And later on, when Parent established the School of Public Health, they had some space in the building. But before they moved in, on the third floor, there was devoted to iron lung patients from all over this area. And 1952 was the height of polio in the United States. There were over 50,000 cases of paralytic polio. And so it didn't take much to energize us to go up and just open the door to that iron lung unit and hear the terrible noise of the motors that kept these things going, and to see these kids and adults in these things. It was inspiring to go back to the lab and work.
It's interesting. I'm sure that would inspire you.
Yes,
right?
it did.
You also worked on the inactivation procedures for
Yes.
the formalin inactivation, right?
Yes. Now, we use formalin because Jonas had, in his work in Ann Arbor with influenza vaccine, he and his mentor, Tommy Francis, had decided to use an activated influenza virus inactivated with formalin. So when we were starting out to make a vaccine, Jonas had the experience of the formalin inactivation of influenza, and what we had to do is adapt it to poliovirus by testing various concentrations and incubation times and so forth. And it would have all been impossible if we hadn't had cell culture.
Right.
Yeah.
Now, I wanted to ask you about the incident right after the release of the vaccine where a number of kids got polio from improperly inactivated. Do you remember that happening in 55, I guess, after the vaccine was licensed? I don't know if you remember that.
Well, I remember it because it's a very painful incident in my life. And it ended my relationship with Jonas Salk. This is in general knowledge, but the time that it was. Right after the announcement of the success of the vaccine in 1955, was in Ann Arbor, and I had to give a paper the next day at a Federation meeting in San Francisco. And so I left Rackham Hall, where the announcement was made, and went to Willow Run Airport and flew to San Francisco. And when I got to San Francisco, I got a telephone call from Cutter Labs, somebody who has the same name as a restaurant I frequently go to before the opera, called Irish name. Anyway, they wanted me to come out and visit them at Cutter Labs, which was in Oakland, because they were having trouble inactivating, having some trouble, and they wanted to get my advice. So I said, well, you know, I'm only going to be here another day. They said, we'll send a car, we'll take you back, we need to talk to you. So they picked me up at the hotel and drove me out to Oakland, and we had a very nice lunch, and then they took me to the plant, and they started to show me their inactivation curves. And what they were getting is virus after they was the asymptotic inactivation, they were still getting positive virus out of infectious virus out of the inactivation batches. And also, they had very bad procedure that they, in a room that we went into, they had a tank that they were growing polio cells in, and inactivating poliovirus. And in another tank in the same room, they had the vaccine ready to be put into vials. And I thought this was a terrible procedure to have these two things in the same room. And it became apparent from their data that they didn't know what they were doing. They were really inept. Well, I went back from that meeting, very much disturbed. And when I got back, Jonas had come back, the hero of the world, and I went to him and told him my experiences in Oakland at the Cutter Labs, and that they didn't know what they were doing, and I think that their vaccine shouldn't be used because I wouldn't trust it to give to my kids. And something should be done about it, and I thought I would write a letter, I would communicate this to the Bureau of Biologics, which would be before the FDA, and to Basil O'Connor, who was head of the National Foundation. And Jonas said to me, I think it would be better and make more sense if I sent a letter and informed them. And I cannot find in the archives any such letter, and he never did it. And I felt personally responsible for being so naive as to trust him to do something. He didn't want to do it because it would have soiled his reputation, which was a high point, and would have made it very difficult for the vaccine for any manufacturer to be issued until more testing was done.
Sure, sure.
And so for the rest of my life, I felt very guilty for being so naive as to think that he would do it. And those 250 kids who died from Cutter vaccine have haunted me to this day.
I'm sure. Were you involved in helping Cutter get their process on track again?
By this time, the fault of the vaccine had already occurred in Western states, and the kids had been paralyzed. And so, of course, the Bureau of Biologics and the National Foundation sent people, but I didn't go there because I was conflict of interest. So I was out of it.
Yeah, that was a dark time in vaccine history because many people think that was the beginning of the anti-vaccine movement in this country.
I think when they always go back to that as an example of the ills and terrors of vaccines.
Right.
So this is my story involved with the Cutter incident and the trust that was broken between me and Jonas.
So how long after that did he leave Pittsburgh?
Did I leave?
No, you always stayed here, I know, but Jonas...
Oh, he didn't leave till 62.
Did you continue to work with him?
Only for a year.
A year.
Until I found that microbiology department was being set up in the medical school in Scaife Hall, and the head of that department was looking for somebody to do cell culture and work with viruses, and people recommended that he try to have an interview with me, and Hubert Bloch, who came from New York, was Swiss. We got along famously, It was only years later that he told me that Jonas Salk had told him not to hire me because I was a terrible troublemaker. So Jonas Salk had his way of getting back at me.
But he didn't, because you ended up here and you've been here ever since.
He failed to interrupt my career.
So you worked on polio for a while after that,
I worked on
right?
polio for a while, trying to do some genetics with poliovirus, making heat-resistant mutants, not temperature-sensitive, but heat-resistant mutants. And I had for all three types mutants that were stable at 50 degrees. And I made the mistake. By this time, Albert Sabin was doing his testing in Russia.
Right.
And I wrote a letter to Albert and said, I got these wonderful, and they're stable, I can pass them, and they don't revert. Wouldn't this be good to have this marker on the vaccine, the live virus vaccine, so you could, the wild type virus has this characteristic,
Right.
and you would be able always to tell whether it was your vaccine or wild type. And I got back the most vitriolic letter from Sabin that nothing's wrong with his vaccine and it doesn't need any improvement and I should mind my own business. He was very forthright.
Many of us received vitriolic letters from Albert Sabin. I have several in my file.
Yeah?
Oh, yes. I would write him and ask him, and he would say, apparently you have never read my papers, or if you did, you don't understand them. Something like that. I'm not surprised.
Speaking of Albert Sabin, I have to tell you an interesting incident. And that is that I happened to be president of the American Society for Virology the year that Albert Sabin was 80 years old, and they were holding at the Fogarty Institute at the NIH an all-day symposium in honor of Albert Sabin, and I was invited to attend this symposium. And Albert Sabin he wasn't well, but he sat there through the whole thing, asked questions. He was the old Albert Sabin, even though he could hardly stand. But after the symposium, the group moved to the Cosmos Club in Washington, where there was going to be a banquet in honor of Albert. And after the sumptuous banquet, we went to an auditorium where various laudatory speeches were going to be made about Albert Sabin. And I was one of the speakers because I had been, as president of the American Society for Virology, I had a speech to make thanking him for his contributions and so forth from the virology community in the United States and from the American Society for Virology. And so I got up, and many other people spoke. When everybody was through speaking, Albert Sabin had the last word.
Of course.
So he got up to the podium and he was talking about the development of his vaccine and giving the history of how he developed his vaccine. And he was describing a meeting at which he announced the results. And he said, and there in the front row was seated John, these were now the highlight people of virology at the time. John Paul, Tom Rivers, Tommy Francis, and he stopped completely short. Stopped. And then he said, Julie Youngner's old boss. Because he couldn't get the words Jonas Salk out of his mouth. And that shows you something about how they felt about each other. The feeling was mutual between them. But anyway, it was an amusing incident.
When I entered the polio field in the 70s, I became aware of the animosity between Salk and Sabin, and in fact saw them at a meeting together, yelling at each other and disagreeing. It was quite animated, and that actually is very interesting in light of the fact that Sabin would never admit that his vaccines could actually revert and cause polio.
Never did till the day he died.
Right.
till the
correct.
day he died.
He always claimed they were wild-type strains that were infecting these people.
He refused to recognize the truth. And it was his ego wouldn't allow him to recognize that, just like Jonas's ego didn't allow him to take my story about Qatar seriously. Both of them had enormous egos and made scientific judgments that were wrong.
You know, as soon as Sabin's vaccines were introduced in 1961, right?
62.
Replacing the Salk vaccines, immediately there were cases of vaccine-associated polio. I don't understand to this day how he failed to see that in all the clinical trials that were done in Russia. They say it was absolutely safe, but it's hard to believe.
I think looking back on it, who saw the data?
Dorothy Horstman, for one, right?
Yeah.
I mean, she's very trustworthy.
Well, but I wouldn't be surprised if the vaccine-induced cases were described as something else.
Perhaps, yeah. So in 2000, by 2000, the U.S. recognized that the risk of the Sabin strains outweighed the risk of wild virus. So we switched back to Salk, and by this time, of course, he had passed away. Did you find that an interesting time in the history of virology?
Absolutely. I found it very interesting that in the United States, first the Pediatrician Association said, well, why don't we give a dose of the killed vaccine and then give the live vaccine? And that became so ridiculously complicated that they finally realized that there's no polio around. They might as well switch completely to the killed vaccine, which is what we use now.
Right.
And all the developed countries use the killed vaccine.
Right. And now, amazingly, WHO recognizes that it has to eventually abandon the Sabin vaccine.
Otherwise, they'll never eliminate polio.
That's right. So here, let me get your thoughts on this. In April of this year, there will be a synchronized switch in 155 countries from trivalent to bivalent Sabin vaccine. So type 2 has been eradicated. So they're saying we don't want to put type 2 OPV, in the vaccine anymore. Nevertheless, type 2 vaccine derived strains continue to circulate.
Yes.
So do you think after we stop immunizing cold turkey, we're not going to even follow up with IPV? Do you think that will be a problem with these circulating type 2 strains?
Absolutely. Absolutely. I think that the ideal solution, which is impractical, because ideals are very often impractical ideas, would be to use the kill vaccine all over the world. I know the people at CDC are working very hard to try to find a concentrated vaccine that they could give in one shot rather than have, but I don't know how far they'll get with this, whether they'll succeed in being able to do this. But the ideal thing would be to use the kill vaccine all over the world, but it's too expensive. It's too complicated because you have to give multiple shots.
Right.
It involves a needle, and not only kids, but the parents object to needles. So the practical solution is impractical.
The other problem is that to produce, so my understanding is that even someday if we eradicate polio, we stop using OPV, we will stockpile IPV in case of an outbreak. But these are produced from wild type strains.
That's right.
Which we're not supposed to have anymore.
Right, I destroyed all mine when they told me to.
Yeah, in fact, I was told last month by CDC I have to destroy all my type 2 strains now. So how do you grow vaccines from strains that you're not supposed to have?
Well, you grow them, you inactivate them, and then you make enough to you store the inactivated vaccine in large quantities. But that's the same problem with smallpox. Same thing with eradication of smallpox, but that's a different story because there are no carriers.
Right. So what a number of labs are trying to do is to make China and Japan have licensed Sabin IPV. So they take the Sabin strains and inactivate them.
And are they as good?
Well, they're antigenically good, but the yields are lower than the wild strains. Yeah. So they're not ideal for production. Other people are trying to make new seed stocks that could be used that would not revert, because the Sabin strains in culture even will revert.
Yeah.
They would be dangerous to grow in large quantities. So that's one of the objectives, is to try to make new IPV seed stocks. So you destroyed all of yours years ago, didn't you? Right.
When I was told to by the CDC.
It's too bad you didn't keep those heat-resistant viruses. We could today sequence them and see where the mutations are. That would be really interesting.
Yeah, well, that would, but
they're gone.
That was not possible when.
Yes, of course. It wasn't possible till the late 70s or 80s. You sure you don't have any of them left, right? Absolutely sure.
I'm not. If I said I did, I would be violating the law.
I'll ask you later. I can ask you later. So do you, right now we've eradicated type 2. There has been no type 3 cases, wild type now, since 2012, and only type 1 remains. Do you think we can eradicate polio?
As long as the oral polio vaccine strains revert, they're going to be carriers. Right. And people can carry that virus in their gut for months at a time. There was somebody who had polio strain, I don't know how many years.
20 years. Yeah.
20 years.
Yes.
20 years after, he was still
Right.
excreting polio virus. So how do we know how many carriers there are of the other strains?
We don't
and
know.
when you have a completely susceptible population, it doesn't take very much to set off another epidemic. So there's a problem in the eradication of polio. And I think that enormous amounts of money have been spent, are now being devoted to continuing to try to get that last case in Afghanistan or Pakistan. And I think I made this statement to a group of people, and they didn't like it. And that was that when you compare the amount of money that's being spent to eradicate the few cases of polio compared to the death rate from measles in the third world.
Right.
Measles is killing 500,000 kids a year.
Right.
And polio now they're trying to eliminate 12 or 15 cases by spending millions of dollars of immunizing large communities, larger communities with the vaccine. And also, the other day there was a shoot-up of a polio unit that was housed somewhere in Pakistan, and they just came in and machine gunned the unit and killed a couple of people. So that's another problem, that is getting that last case, if they ever can, is going to be difficult. And in some ways, it's a misuse of resources. TB, malaria are killing millions. So the elimination people don't like to hear that said, because they're so devoted to the eradication. The suffering that other diseases cause so outweigh the polio that somebody has to make some decisions at some time.
I think it's been harder than they envisioned.
That's right.
Right. The original goal was 2000 for eradication.
It was even before that.
It was before.
They thought it was 89.
So they didn't recognize this. And once you start this kind of a program, if you stop, it's a big problem, I think. I think we're also fortunate that the Gates Foundation has funded a lot of the vaccines.
Yeah.
And I think he's committed to seeing it through to the end. But I do think these circulating vaccine-derived viruses are a big problem. And I don't see the solution unless we just do an experiment and stop, which is what we're doing in April. So we'll see what happens. I just suspect there will be polio afterwards.
Yeah.
We'll see.
It's a hard road there.
Yeah.
Right.
So did you stop working in the lab many years ago?
No. No, I continued to work in the lab till I closed the lab, I don't know, in the 90s. Mm-hmm. In the 90s.
And
I worked on influenza.
Influenza, because you stopped working on polio in the 60s,
No, I worked on
right?
interferon for a long time. Actually, my lab was first to find gamma interferon, which is we call type 2, but it was different than.
So the interferon was discovered in the late 50s?
Late 50s.
So you must have seen that and picked up immediately on it, right?
Well, it's an interesting story how I got into interferon research. I was working on influenza, doing some kind of genetic experiments in culture with influenza. And I went to a seminar where a group had been working on interferon. Mm-hmm. And they said, in the seminar, they said that interferon is designed by nature of the cells to combat viruses. And after the seminar, I started thinking, that's pure teleology, pure. And I can't stand teleology. So I thought, what if there are other intracellular organisms or substances that could produce interferon? So it was Warren Steinbring was working with Brucella abortus, and so he had concentrated cultures of Brucella abortus inactivated, and so we injected. I had the chicken working with influenza. I had chicken monolayers growing all the time, chicken monolayers, and so we injected intravenously Brucella abortus into chickens, and then bled them few hours later, or a day later, so forth, and then did a test in chick embryos. Because the description of interferon by Alec Isaac is it was species-specific. So we had the species-specific host and the test. And it inhibited VSV. We published, then we found that you didn't need bacteria. You have to use endotoxin.
Right.
And so that was the first example that interferon could be produced by non-viral substances. Then other people found double-stranded RNA would do it too. And that was interesting. But as an aside, Alec Isaac in England, who was the discoverer of polio, never invited me to any interferon, never invited me to any of the interferon meetings in England that he held every year, because he never would admit that something other than viruses could induce interferon.
Yeah, that's great. Now we know so many things can induce interferon. So this was just the very beginning of learning this.
So that's how I got into it, because of my disgust for teleology. And so I went on from there. We found what we call type 2 interferon, which is now gamma interferon. And that was another big contribution from my lab in the interferon field.
I also noticed you had some papers you were studying something produced by vaccinia virus that inhibits the PKR kinase.
That was the first example of a virus that produced an anti-interferon effect.
Right, exactly. And now we know exactly what these gene products are, and people have their structures and their mechanisms. I think that's very cool that you were
We knew that it was the kinase it was inhibiting. That's as far as we could go. And we knew it was EIF2 that was was involved. And but we couldn't go any further.
And now you know these proteins are EIF2 alpha mimics
Right.
that
right.
distract the kinase.
Right.
But I think this is beautiful. You've got so many seminal things here that today we take for granted, the different kinds of interferon.
Nobody remembers any of this. You know, it's interesting. It's interesting. Unless you make a discovery like in 10 years, people forget the past.
Of course. I had a professor in grad school tell me, Vincent, it doesn't matter what you've done, it's what you're going to do.
That's right.
People just forget it, but it's all in the literature.
Exactly. So I was also fortunate to be working in the lab and having a group with me all through the golden years of NIH funding and Army and the military funding. And it was something that couldn't happen today. I had the same R01 grant number for 22 years. So today that couldn't happen because they make you do something they won't do. You have to continue to be creatively ahead of the study section.
Yep. So you're saying there's a problem with science today?
Yes, there is. There is a problem with science today. First of all, do you have enough time for this? You notice I haven't looked at my watch.
Don't worry. We have time. If people want to leave, they're welcome.
Of course, Vince told me before we started, I asked him the question whether he does the cutting himself, and he said, yes, he does the editing himself. So anything I say, he could edit out and you'll never hear it again.
No, I won't edit it, no. I only edit the beginning and the end. In the middle, it stays.
Okay.
Everything.
So they're not clippings on the floor of Youngner talking.
No clippings. What's wrong with science?
What's wrong with science is that there's been a move away from the funding and interest in individual scientists and their ideas. And I think the programmatic funding of research by NIH and others has really set back science rather than advanced it. People disagree with me and say, look at all the great things, and I say, what great things? A lot of the connection between the university and industry is also taking away from the individual scientist and their creativity. And I think that's where all the good ideas and all the advances come from that we can enumerate is from the individual creative mind of a scientist, not from a program director who is overseeing a multi-institutional, multitasked project which sucks the money out of the R01 fund.
Is this likely to change anytime soon?
I don't think so. I think that they do lip service to the R01s, but unless there's some amazing prosperity and glut of funds, no deficits, and no programmatic forcing by Congress to study one disease or another because either they have a relative who has it or they're afraid of getting it.
Yeah.
That's why we have so much Alzheimer's research, is because the Congress are aging and they want some cure quickly.
I happen to agree with you. I think that the best things in science come from letting individuals pursue their curiosity. So if you were young today, or if there are many young people who want to go into science, what kind of advice would you give them? Would you tell them to stay out of the field entirely?
Well, I think, I don't know what the, microbiology has now resumed importance that it had lost somehow back in the 80s. It really has come back to its own, in its own. And I think that a lot of the advances that are going to be made are going to be through microbiology and molecular biology. And I think that young people today have to think about what is their career going to be. Academic careers are becoming harder and harder to find. Tenure in many institutions are harder and harder to obtain. So what does that leave? Leaves research institutes and pharmaceutical and other scientific institutes. But I'm not saying this to discourage anybody, because if you have good ideas and study sections are sympathetic, I think you can make a career in science on your own. But it's tough. It's getting tougher.
So if you were starting today, you would still do it again?
I would do a lot of things over again, and some things I would hope I had never done.
Right. Do you miss doing research at the bench?
When I shut my lab, the last gasp of my scientific career was getting to produce the second vaccine ever produced at the University of Pittsburgh. There were only two, polio and the cold-adapted equine, attenuated equine influenza virus, which is now on the market. So the last gasp was helping horses too. So you can't say, PETA would give me four stars.
Okay. Is there anything that we missed we should cover?
I think you've exhausted me.
I have one more question. This is the hardest question of all. I saved this only for special people. If you could have dinner with anyone who is living or dead, who would it be?
I think that I would like to have dinner with President Obama. Obama is a unique person and has been a unique president. And if someday in the future in somebody's biography of Barack Obama, they have a footnote that on such a date in 2000 and something, Julie Youngner had dinner with Obama, it would be the greatest tribute that I could find.
That's great. You know, I understand that he's going to be a professor at Columbia after he steps down, okay? And that's, of course, where I am. So I'm going to have him as a colleague, and I'm hoping to get him to take my virology course. Or at least come to one lecture, and then my daughter, of course, would want to be there at the same time. That's great. I like that answer. Very good. And you didn't even have to think very long.
No, I didn't.
So this episode of TWiV will be like all the others at iTunes, and you can also find them at microbe.tv/TWiV. And we love to get your questions and comments. Please send them to twiv@microbe.tv. I want to thank my guest today, Julius Youngner. Thank you so much for talking with me here at the University of Pittsburgh. I also want to thank the department for inviting me. So tomorrow I hope you come to the Julius Youngner lecture, which I'm giving.
I wouldn't miss it for the world.
That's an honor for me to do that. I want to thank the department for inviting me, and Carolyn for inviting me. Where's Carolyn? Is she here? Hi, thank you, Carolyn, the shy Carolyn. And the students and postdocs for asking me to come. And I was very happy to do this. I'd never been to Pittsburgh, as I said, so thank you so much for doing this. And I want to thank the audience for sticking around as well. I hope you enjoyed it. I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. exhausted, really? We could do another one. Anybody else want to?